Sequence bioinformatics. Genetic data represent a treasure trove for researchers and companies interested in how genes contribute to our health and well-being. Almost half of the genes identified by the Human Genome Project have no known function. Researchers are using bioinformatics to identify genes, establish their functions, and develop gene-based strategies for preventing, diagnosing, and treating disease. A DNA sequencing reaction produces a sequence that is several hundred bases long. Gene sequences typically run for thousands of bases. The largest known gene is that associated with Duchenne muscular dystrophy. It is approximately 2.4 million bases in length. In order to study genes, scientists first assemble long DNA sequences from series of shorter overlapping sequences. Scientists enter their assembled sequences into genetic databases so that other scientists may use the data. Since the sequences of the two DNA strands are complementary, it is only necessary to enter the sequence of one DNA strand into a database. By selecting an appropriate computer program, scientists can use sequence data to look for genes, get clues to gene functions, examine genetic variation, and explore evolutionary relationships. Bioinformatics is a young and dynamic science. New bioinformatics software is being developed while existing software is continually updated. The most important thing in sequencing is the identification of the gene within the given DNA sequence. Once the sequence has been assembled, bioinformatics analysis can be used to determine if the sequence is similar to that of a known gene. This is where sequences from model organisms are helpful. For example, let's say we have an unknown human DNA sequence that is associated with the disease prostate cancer. A bioinformatics analysis finds a similar sequence from mouse that is associated with a gene that codes for a membrane protein. It is a good bet that the human sequence also is part of a gene that codes for the same membrane protein. In such a way, the bioinformatics sequencing can be useful to find an unknown gene or protein of interest. There are bioinformatics programs like ClustalW, BLAST, etc. are available for sequencing. One of the widely used databases for sequence data is UniProt. The Universal Protein Resource, UniProt. The Universal Protein Resource is a comprehensive resource for protein sequence and annotation data. UniProt is a collaboration between the European Bioinformatics Institute or EMBL, EBI, the SIB Swiss Institute of Bioinformatics and the Protein Information Resource. Across the three institutes, more than 100 people are involved through different tasks such as database curation, software development and support.